Assalamu alaikum guys. Continuing from where we left in the previous episode, we will be talking about branches of microbiology and microbiology pioneers in this episode. So branches of microbiology, we have firstly the microbiologist, which is the scientist who study microbes. We have the bacteriology, which is the study of bacteria, mycology, the study of fungi, protozoology, the study of protozoa, phycology or egology, the study of algae, virology, the study of viruses and prions. Notice that mycology and phycology has similar names, so make sure you can differentiate between the two because they might be tricky. Okay. General microbiology is the study of microorganisms as a biological system. This is the field that we are studying right now. Medical microbiology is the study of pathogens and infectious diseases related to humans. Veterinary microbiology, veterinary means veterinary, so this is the field that studies pathogens and infectious diseases related to animals. Sanitary microbiology, they study the microbiology that is related to public health. Sanitary means صحي. Agricultural microbiology, agricultural زراعي, studies the microbes of microbes in soil and fertilization of plants. Industrial microbiology studies the microbes used in manufacturing products like alcohol, acids, yogurt, cheese, etc. etc. Environmental microbiology, ecology, studies the relationship between microbes and the environment. Microbial physiology and genetics, lastly, is the field of microbiology that studies microbes individually, how they act, how they live, how they multiply, and what's their genes. Okay, moving on to microbiology pioneers, some history, brief history about these scientists. So firstly we have Anthony van Leeuwenhoek. This guy was the first, maybe the first human, it is questionable, but he was the first one who saw living bacteria and protozoa in pond water and scrapings of his teeth. He used a simple handheld microscope, single lens with 300 times magnification power to see living bacteria and protozoa. He is considered the father of microbiology, bacteriology, and protozoology. Louis Pasteur, or Louis Pasteur, I guess, he was a chemist, a French chemist. He was ordered to investigate the cause of spoiled wine. So wine with time, its taste change and becomes more acidic. While studying why that happened, he discovered that yeast is a living material. Yeast is well known for humans, but he was the one that discovered that this is living material. And he discovered that this a type of yeast is responsible for wine fermentation by converting sugar into ethanol. Also, he discovered that other microbes are responsible for other fermentation products like vinegar. Vinegar is produced by converting ethanol into acetic acid. He discovered that this conversion is done by a microbe, a type of microbe. We now know that this type of microbe is called Acetobacter, a type of bacteria. He discovered that aged wine. Now moving on to the topic, he was ordered to observe and find the reason why. He discovered that aged wine is spoiled because of a different microbe that converts sugar into lactic acid. We now know that microbe as lactobacillus acidophilus. He discovered that heating wine to 55 Celsius for a few minutes was sufficient to kill the microbes and prevent spoilage with time. So he used heat to kill microbes, That way he knew that the reason for the convergence is microbes, because when he used the heat, no spoilage occurred. Okay. This process of heating things to kill the microbes is now called pasteurization, um, uh, to honor Louis Pasteur. Today we use pasteurization for milk and other dairy products by heating milk to 73 Celsius for 15 seconds or to 63 Celsius for 30 minutes. This is done in order to kill the pathogens. Now, pasteurization only kills pathogens, but not all the microbes. Louis Pasteur also we value him so much because he developed multiple vaccines, especially animal vaccines. He developed vaccines for rabies, chicken cholera, anthrax, and skin erysipelas. And he was the one who tried rabies for the first time. He had the courage to try rabies vaccine on humans. He tried it on a 9-year-old boy who was bitten badly by a dog, and he prevented rabies from developing in that boy. He showed the importance of hand washing and championed changes in hospital practices to minimize the spread of disease. So he is a chemist, but he advised physicians of the importance of hand washing, and he minimized by that the spread of disease. He introduced the terms aerobes and anaerobes. Aerobes are the microbes that require oxygen for survival. Anaerobes are those that don't require oxygen for survival. Louis Pasteur also made significant contributions to the germ theory of disease, which says that specific germs cause specific infectious diseases. How did he contribute to that theory? He discovered that the silkworm diseases that infected the silkworms in France, silkworm dut al-qaz, is caused by a type of protozoa. So he discovered the infectious agents that cause silkworm disease. And this helped the germ theory of disease to be proven. He disproved the theory of spontaneous generation, or abiogenesis, proving in turn the theory of biogenesis that was proposed by Rudolf Virchow. So there was a theory that said life can arise spontaneously from non-living material. This theory is called abiogenesis, or spontaneous generation, النشوء التلقائي, and he discarded that. How did he do that? Well, he did a simple experiment using a swan neck flask, قارورة عنق البطة. This type of flask, he put a liquid inside. The liquid is heated. The heat should theoretically kill any microbes. So when he observed the bacteria afterward, the liquid afterward, after heating it, no bacteria was present. But when he heated the liquid, then removed the neck and allowed the air to enter and all the contamination in air, the bacteria was present. So the bacteria did not spontaneously originated from the liquid, but rather come from outside. Also, he did he heated the liquid, allowed the flask to tilt sideways so that the liquid can touch the walls of the flask. Okay, the neck. Now the walls might have some microbes. So when he tilted the flask sideways and allowed the liquid to contaminate with the walls, the bacteria was present, although he heated that liquid. So he discovered that bacteria can come by air from outside or from other parts inside the flask, but not originate from the liquid itself. So he said, never will the doctrine of spontaneous generation recover from the mortal blow of this simple experiment. There is no known circumstance in which it can be confirmed that microscopic beings came into the world without germs, without parents similar to themselves. Okay, lastly, Louis Pasteur created Pasteur Institute and its international network as clinics for treatment, research, and teaching globally. One of Pasteur's students, Alexander Emily Jane Yersin, the director of Pasteur Institute in Vietnam, discovered the causative bacteria of plague, Yersinia pestis. Now, moving on to Robert Koch. This is a German physician. He developed methods of fixing, staining, and photographing bacteria. He could grow bacteria on potato slices, so he developed methods of cultivating bacteria on a solid media. He used petri dish with agar to obtain pure cultures of bacteria. Firstly, petri dish is a flat, round glass developed by one of Robert Koch's colleagues called R.J. Petri to culture bacteria on a solid media. While agar is a polysaccharide obtained from seaweed algae, suggested by another Robert Koch's colleagues, Frau Hesse, as a solidifying agent. So he used these two parts together to obtain pure cultures of bacteria. Now cultures of bacteria are growth of bacteria. Now this growth might be of multiple types of bacteria, while the pure culture of bacteria is the growth of single type of microorganism or a bacteria on a culture medium. So he was able to obtain the pure cultures. Now, Robert Koch is the one who proved the germ theory of disease. Now, we said that Louis Pasteur contributed to the germ theory of disease by discovering the causative agent of silkworm diseases, but the one who proved the germ theory of disease is Robert Koch. He proved that Bacillus anthracis is the cause of anthrax. He discovered Vibrio cholera as the cause of cholera. He discovered Mycobacterium tuberculosis as the cause of tuberculosis. He also developed PPD test to diagnose tuberculosis. Now, the major contribution of Robert Koch, okay, how could he prove the germ theory of disease and prove all these things? How could he do that? He put forward the Koch's postulate, افتراضية Koch, for identifying microbial cause of a disease and proving the germ theory of disease. So what's this postulate all about? Firstly, a particular microbe, okay, must be present in all the cases of the disease and must not be present in healthy ones. So if we have this sick rat and multiple sick rats, we should find a particular microbe in these sick rats that are not present in healthy rats. Secondly, this microbe must be isolated from the disease and grown in a pure culture in the lab. So we take this microorganism, use Koch's ways of fixing, staining, growing, etc., to obtain a pure culture of this microbe in the lab. Okay. Then, when microbes from pure cultures are inoculated or injected into healthy rats, healthy animals, the same disease must be observed. The same disease, that the rat will become sick from that microbe that was found in the sick rat in the first place. Then this sick rat, the experimentally infected sick rat, must have the same microbe that was recovered here, and we must be able to re-isolate the microbe from the experimentally infected sick rat. Okay. Now, Koch's postulate, okay, is true for most of the microbes. most of the microbes follow the Koch's postulate, which means if we can take the microbe from any sick case, then grow it in a pure culture, okay, then inject it in an animal, any animal, okay, then the same animal will become sick, and we could be able to take the microbe, regrow it, and re-isolate it in the lab. Now, some microbes don't follow Koch's postulate. Why? Because some microbes are, for example, obligate intracellular pathogens. They only live inside tissues. They cannot be grown in vitro in the lab. Examples of those obligate intracellular pathogens are all the viruses and some bacteria. Some bacteria like Chlamydia, Rickettsia, Mycobacterium leprae, the cause of leprosy, and Treponema pallidum, the cause of syphilis. So these 4 types of bacteria, you have to remember them by heart. These are the obligate intracellular pathogens, along with all the viruses. Now, second reason why some pathogens don't follow Koch's postulate is because these pathogens are fastidious. What we mean by fastidious in Arabic, hassas. So these microorganisms require specific nutrients that are not found normally in cultures. Why they are not found in cultures? Well, because most of the time we don't know what they need. So an example for them are Treponema pallidum also. So Treponema pallidum cannot be grown in the lab for 2 reasons. First, it is obligate intracellular pathogen. Second, it is a fastidious microorganism. Thirdly, there is something called species-specific microorganisms. These are microorganisms that can only infect one species. For example, we have humans pathogens. They only infect humans. So when we isolate them, okay, then inject them into animals, these animals will be healthy. They will not acquire the disease, because the microbe that causes that disease is only causes disease in humans, but not in animals. Lastly, we have what we call synergistic or interactive infections. Those are caused by more than one microbe, like bacterial vaginosis. Bacterial vaginosis is caused by multiple microbes, not just one microbe. Okay, and that is the end of chapter 1. Thank you for listening. Please share, subscribe, like, do everything, and be tuned for the next chapter. Thank you. Assalamu alaikum.